speaker is Jim Hafernik. Dr. Hafernik is a professor of entomology emeritus from San Francisco State. He sort of discovered phorid flies in the area when some hatched out in a vial on his desk a number of years back. And shortly after that, he did a first run of this program for our club, visiting us when we were over in the Woodbury Nature Center. And now he's consented to give us a recap and an update on phorid flies in our area.
All right, welcome, John.
Well, thanks. Thanks for the introduction, Jerry, and thanks to the group for inviting me back. I think that was about 2013 or something like that was the last time I was here. Had a good time, and I look forward to talking to you tonight about some of the new things we found, and I'll give you a recap of some of the history of studying phorid flies in the Bay Area and beyond as well. Let's see. All right. So what I'd like to talk to you tonight about is this phenomenon of phorid flies parasitizing honeybees, which is something that's a relatively recent phenomenon, at least in terms of being recognized in North America. It's been something that's been happening in South America before. And it's something that, as Jerry mentioned, I discovered some years ago back on the San Francisco State campus. Okay, so first some information about phorids. Phorids are a big group of flies. There are about 4,000 described species, maybe as many as 8,000. Brian Brown, who's the expert on phorid flies at the Natural History Museum of LA County, made a bet with one of the trustees of the institution that if he set out flight traps, these are traps designed to catch flying insects, especially small flies, in this fellow's backyard, he bet him he could find a new species of phorid within a few weeks. And so sure enough, he did that. And Brian indeed did find, I think, more than one new species of phorid in the backyard of this trustee of the LA County Museum in metropolitan Los Angeles. So you don't have to go far to find new species of these creatures. You probably have undescribed species flying or scuttling around your backyard. In fact, sometimes they're called scuttle flies because rather than flying a lot, they often land and sort of have a nervous scuttling-like behavior on leaves or horizontal surfaces. And most of the time they're overlooked. Sometimes they're also called humpback flies because they have a kind of humpbacked appearance. And they also have these dark veins at the base of the wings that are typical of phorid flies. They're, as I said, a large group, so they do a lot of different things. Some are scavengers, probably the best known ones are scavengers. Some are herbivores, some are predators, and the ones we'll be most interested in tonight are parasitoids. Parasitoids being parasites that actually kill their host, rather than eating on them slowly, they eventually completely eat the inside out and kill them. Some of the more famous cast of characters in the Phoridae, the coffin fly, you can imagine how it gets its common name. It's a fly that's often found in decaying bodies, feeding on the decay of human bodies and other bodies as well, distributed widely around the world. And it's one of those CSI kind of creatures that you might use for crime scene investigation of determining time of death and things like that. As I said, phorids are small flies. The biggest phorids, maybe 4 millimeters or so. So that's less than 2/10 of an inch. And in fact, one of the points of honor of the Phoridae is that it contains the world's smallest fly with one of the world's largest names for a fly, Megacelia arnoldi, named for Arnold Schwarzenegger, because if you look at the underside of this fly, its legs perhaps look a little bit like the muscles that Arnold once had in his younger days. It's a small fly, only 3/10 of a millimeter, so that's about 2/100ths of an inch in length. It's flightless. Don't know for sure what its life history is, but other similar kinds of flies are parasites, parasitoids on very, very small ants. So even if you're really small in this world, there's a phorid fly that can get you. And this is the world's smallest fly. For comparison purposes, the world's smallest insect is a Hymenoptera, a small parasitic wasp called the fairy wasps. And these are wasps that develop in the eggs of other insects, especially aphids and things of that sort. Probably the best known phorid fly is Megacelia scalaris. It's a part of the largest genus of phorids, which includes over 1,600 species, with new species being described all the time. It's a scavenger found all over the world and sometimes a facultative parasitoid. Indeed, it sometimes shows up in living honeybees and often shows up in dead and decaying honeybees. Probably the most famous parasitic phorids are those that attack fire ants. And in fact, they're so good at attacking fire ants that they've been imported from South America, where fire ants are native, into the southern US, where fire ants are an introduced pest, to try and control fire ants. Here's a phorid on its way to attacking a fire ant, lay an egg inside. Eventually, the single maggot will eat the inside out of the ant, push its way up into the head, and the ant, before it dies, will wander out of the hive, kind of like a zombie. Sometimes they're called zombie ants, and die away from the ant nest. Now, one of the interesting things is that these creatures are often called decapitating flies because as the mature larva emerges from the ant, it pops off the head capsule of the fly, of the ant, and to add insult to injury, even pupates in the head capsule. So here's an adult fly emerging from the head capsule of one of these fire ants to go on to look for more fire ants to parasitize after it finds a mate. Now, there also are phorids that are brood predators and also sometimes kleptoparasites. You've all heard of kleptomaniacs, shoplifters. Well, kleptoparasites are parasites that come in and steal the resources of another organism. In this case, the pollen stores and other things, and mostly stingless bee nests in South America and Central America. And they'll also, in this case, feed on the larvae in those nests as well. So these are the maggots, the larvae of this particular fly, Pseudohypercera curtisii, the most famous of these kleptoparasitic phorids. They're a major pest of stingless bees, and stingless bees are commercially produced in Central and South America for their honey, as well as honeybees in those areas. But the native bees culture goes all the way back into the native cultures of Central and South America. And that still goes on today. This is a pretty nasty pest of that native culture. They're also sometimes found in honeybee hives, where they feed again on the pollen and sometimes on the brood. Weakened hives especially are vulnerable to being invaded and eventually to collapsing because of these creatures. Now, I mentioned Megacelia before. A number of species of Megacelia have been found in honeybee hives. Several species in the Bay Area and a number in Europe, North Africa, the Mediterranean area, and so on. Usually these are scavengers on dead or dying bees, larvae or pupae. If you have a failed hive, it's not unusual to have Megacelia come in and feed on the dying remains of that hive. And you may find Megacelia larvae and pupae in the hive. You may also find them in dead bees that are distributed around the hive as well, or even away from a hive at some distance. There have been some interesting studies, though, recently in Europe, the Mideast, and Africa that have found Megacelia larvae parasitizing living bees. So we sometimes say that they're facultative parasites. They can live as a scavenger, but they can also live as a true parasite inside a living organism. And that seems to be the case occasionally. Mostly we think this happens with bees that are already in pretty bad shape, that are either diseased or dying or on their way out. But they've also rarely been found now in Italy in bees coming to lights at night, where researchers put out light traps in a way that I'll talk to you about in the Phorids that we study in the Bay Area, attracted some Megacelia and found, attracted honeybees and found some Megacelia, and some of them attracted to lights. Megacelia is really a pretty amazing creature. Megacelia scalaris not only can feed on decaying honeybees, it can feed on decaying humans. Sometimes it can get into open sores, and it's even been recorded feeding on latex paint. So virtually any organic substance is something that can produce a Megacelia. You go down into Central and South America, there's a group of bees that have the wonderful common name, the bee-killing flies, Melaleuca. And this is a group that parasitize native bees, including those stingless bees, again, that are found in Central and South America and used for honey production down there. There are at least 8 species of this genus that have been recorded parasitizing honeybees. Brian Brown tells me that he's seen a number of other species laying eggs on honeybees, although he doesn't know if the larvae have survived on them or not, but he suspects that they probably did. They can cause pretty high mortality in bees in Brazil, where they'll infect both the domestic and Africanized honeybees. These bees use their ovipositor in the female. You can see the ovipositor here. When you see a Phorid with a long ovipositor like that, you can be pretty sure that it's a parasitoid that's going to insert its eggs into something. And in this case, it's in the bees, and it's going to find a weak spot. The weak spot on a bee is going to be the membranes between the abdominal segments, the abdominal plates that are on the abdomen. Sticks that hypodermic needle in, injects eggs, the larvae hatch, and then as the bee dies, they eventually feed on its thoracic muscles and most of the inside of the bee before emerging from the junction of the head right up here as maggots and slurking off to pupate away from the host. Sometimes in South America, I'm told that hives that are infected by Melaleuca are said to have a disease called autumn disease, because it tends to be something that happens during our springtime, their autumn down in South America. Something we need to be aware of is that as the climate changes and warms, there is certainly the potential of some of these creatures expanding their range into the US, or even today being brought in accidentally through commerce of various sorts. Interestingly, there is a report of a colony of stingless bees now living in Palo Alto that I think a young kid in Palo Alto found a short while ago. And it's something that was introduced by a researcher back a number of years ago and apparently still survives in Palo Alto. So there might be some native hosts for these bees if they found their way into this area for these flies. But the main show is this species, Apocephalus borealis, a creature that's now been called a zombie fly in a variety of different venues, mostly the press, but occasionally in entomological publications as well. It's one of 343 known species of Apocephalus. So this is number 2 in species diversity in the Phoridae, next to those scavengers that we talked about, the Megacelia. And many of these species parasitize ants in Central and South America. And they also get their name Apocephalus from the fact that they'll sometimes pop the heads off of these ants when they emerge as larvae. There are a variety of species that are found in middle elevations of Central America. And most of them we know nothing about in terms of their hosts or what their biology is like. Some of them closely resemble Apocephalus borealis that we have here in the Bay Area. And the suspicion is that they probably have similar habits of parasitizing Hymenoptera, although some species have even been recorded from lightning bugs or fireflies. The native hosts for this particular species, and it is a native fly to North America, includes bumblebees, yellowjackets, and a variety of other Hymenoptera, and perhaps even some spiders. Again, it's a small fly, so it's going to be smaller than a fruit fly. It's pretty stealthy, often not noticed. And this is the fly that I discovered on the San Francisco State campus in honeybees that were stranded under lights. And it turned out to be parasitized. And the bees showing zombie-like behavior, flying out of their hives, in this case at night, perhaps during the day as well, but certainly at night, and being attracted to lights. And you can see this creature also has a very nice ovipositor and has a life cycle very similar to the killing bees that we talked about in Central and South America. Basically does pretty much the same thing in terms of the way it kills its host. Now this almost brings a tear to my eye now that I've been retired for a few years. This is Hensel Hall on the San Francisco State campus, a place that I spent 40 years of my life as a biology professor. And one of the things I'm most proud of during those 40 years is that I never purchased a parking permit. A lot of conflict on campus over who gets the best parking, and I didn't want to be a part of that. So I'd always come in early, and I'd park on 19th Avenue, which is sort of back down this way and runs in front of Hensel Hall. Get there early to beat the students who'd also try and find those parking places. And then I'd always walk into the front door of Hensel Hall, which is right here under this shadow area. And some years ago, I started noticing that as I was walking in here on the concrete sidewalk area, there were a number of bees that were either dead or dying, some with their tongue sticking out. And it's one of those serendipitous discoveries that's so typical of science. We think of science as being so carefully planned, but in fact, a lot of important discoveries are made by accident rather than by planning. And this certainly was an accidental discovery for me. I mean, but when I looked at it for a while, I immediately had that kind of a eureka moment that you get when you've discovered something you haven't seen before. And that thing that flashed in my eyes and into my brain immediately was mantis food, which is probably what you were thinking already anyway. Mantis food because I just come back from a field trip with a bunch of entomology students. We went over into Del Porto Canyon in the Coast Range and had brought back a number of rather hungry live praying mantids that needed something to be fed to and something to eat. And mantids often eat bees and things that come to flowers in nature. So I thought, wonderful, mantis food. Started scooping them up in a vial, bringing them down, feeding the mantids. All was good with the world. The mantid loved them. Honeybees, not so much. And all was right with the world until one day I had one of those senior moments, probably why I had to retire, and walked up to my office and instead put one of these vials down on my desk and left it there for an unknown period of time. Some days later, I looked at it and suddenly noticed all of these little fly pupae that were in that vial. And I immediately knew that something was going on inside those honeybees. Wasn't sure what. Waited for the pupae to emerge and produce flies, recognized them as phorids, but really didn't know that much about phorid flies, but knew that Brian Brown at the LA County Museum was a phorid specialist. So I called Brian up, sent him examples of the flies. He recognized them immediately, said they were Apocephalus borealis, a species he'd published on, that they were known to be parasites of bumblebees, but he'd never heard of them coming out of a honeybee before. With that information, I got on my phone again and I called up Eric Musson at UC Davis Extension, the great bee entomologist who is now retired also, and asked Eric if he'd ever had any reports of phorid flies and honeybees in California. And he said, no, he hadn't, but he'd been getting these reports of unusual behavior in bees. People reporting bees coming to their porch lights or to their driveway lights. And maybe were these bees that I were collecting being attracted to lights at night. I said, oh, that's a good idea. hung up, went outside, looked up, and looking before, mostly when I'm walking in urban areas, I look down towards the sidewalk, never know what you might step on. And there was the answer. The bees were coming to the lights that illuminated that overhang area in Hensel Hall. You can see in this picture that a number of them have gotten caught up in the light itself, and there are a few that are still trying to do what bees do around lights. So where were they coming from? Well, they were coming from a hive that Gretchen LeBoon, my colleague at San Francisco State, had placed near the base of Hensel Hall. And the lights come on at night, the ZomBees, the parasitized bees, had a beeline straight up to that bank of lights. And then would, in the morning, you'd find them under the lights, scurrying around in pretty bad shape. Some of them had already died, and many of them died of a few hours later at most. Here's an example of one of those bees and the kind of behavior that you see often in them, this sort of disoriented, circular behavior that gives some hint that there's some sort of neurological problem going on with them at this point. Sometimes you see this in downer bees that are just on sidewalks and various places, parking lots, etc., away from their hives, showing this kind of behavior as well. And sometimes those downer bees are parasitized. So here's a female Apocephalus borealis infecting a worker honeybee. And you can see she also has found that sweet spot, that membrane between the segments of the abdomen of the honeybee, inserting eggs, and in an instant, she'll insert probably several eggs into that bee before it can do anything about it. And you can see the bee kind of looking over its shoulder, not very happy. All of this happens in an instant, and this is a marvelous photograph taken by Chris Kwok, one of my collaborators and a former master's student at San Francisco State, who took this remarkable picture. You have to be there to appreciate it, the speed that all of this happens, the difficulty of getting things in focus, and he nailed it. So here's what's going on and the kind of speed that it takes. You'll notice that poor honeybee had probably 3 or 4 females on her at that point, laying eggs into her. So she probably came out of there with a dozen or more eggs in her abdomen, each one that can produce a maggot, each one that can go on to produce another zombie fly. Once they mature, they move forward and eat the thoracic muscles, just like those other bee-killing flies. And then they emerge at the junction of the head and the thorax. And you see these maggots kind of billowing out here. 1, 2, there might be a third one back in here coming out on their way to find a place to pupate. Everybody have a good dinner tonight? If you have kind of a squeamish stomach, you may want to get ready for the next slide after this one. They do occasionally knock the head off a bee. And you can see this bee had several maggots coming out of it. One of them actually pupated inside the head capsule, as sometimes the ant parasites do. But in most cases, this doesn't happen. This is less than 10% of the time that they knock the head off. And rarely do they actually pupate in the head capsule of the bee. Okay, this will slowly come into focus. So if you're kind of squeamish, you can look away. Here are the maggots emerging from the juncture of the head and the thorax of this long dead bee. So it's feeling no pain at this point. And they will go on, squeeze their way out, slither away, and find a place nearby to form their pupae that will go on to metamorphose into adult bees, or excuse me, adult flies. And you can see they have this rather slow kind of loop-like movement as they go around. And they can travel some distance. I've had vials like this come open on my desk and later find fly pupae at some distance scattered around my office. So Robert, this might be your photograph, if I remember correctly.
Yes, that it.
Yeah, it's an example of how many pupae, how many larvae can come out of a single sample of honeybees in an infected hive. This is a remarkable shot, and it's really a quite artistic shot as well. I really like it, and it shows that in heavy infestations, a single bee can produce as many as 18 pupae. And in our lab, we've been able to infect bees and get out as well over 20, 21, 22 or more pupae out of a
John, I
single
think
bee.
I have the record of 25.
25. Well, okay.
Yeah.
I'll give you that one. Pretty remarkable. Think of how many they could get out of a bumblebee. But 25 out of a honeybee is a pretty remarkable thing. And it shows how efficient these flies can be at reproducing. There's an advantage to being small. You can turn over quickly, produce a lot more individuals quickly. Same is true of the COVID virus, unfortunately. Small things, quick turnover time, potential exponential growth. So as I mentioned, the zombie fly is a native fly, and it has a number of native hosts. And this is one of the local bumblebees that's a common host of Apocephalus borealis, the yellow-faced bumblebee, Bombus vosnesenskii. And here you can see this is kind of an acrobatic Phorid doing look ball, no hands, I can still oviposit kind of dance on the back of this poor bumblebee. Sometimes in the Bay Area, we found as many as 80% of foragers late in the year, September, infected by these flies. So they can do a job on bumblebees, but of course bumblebees survive as overwintering queens rather than as foragers. So the loss of a forager at the end of the year for a bumblebee isn't as big a deal. But queens get hit occasionally as well. Here's some of the other hosts of Apocephalus. Heard some people talking about yellowjackets. So if you don't like yellowjackets, then Apocephalus borealis is a beneficial insect because it's a parasite of yellowjackets as well as bumblebees and now honeybees. Here's a yellowjacket trap, and you can see an Apocephalus pupa that's come out of some of these yellowjackets that were caught in the trap. We also found it in these little potter wasps. This is a solitary wasp that makes a little mud nest. Here's some of the typical Apocephalus pupae coming out of it. A colleague of Brian Brown has found them in Xylocopa and carpenter bees down in Santa Barbara area of Southern California. Chris Kwok has found them in Anthophora bees on the San Francisco State campus. This is a ground nesting solitary bee. And there's a record in the literature of them coming from black widow spiders, quite a different kind of host. But we've tried to infect black widow spiders. The flies kind of show interest in them, but they don't seem to be able to actually penetrate the skin of the spider and lay an egg. Spiders don't have those membranes on their abdomen. They have a completely hardened abdomen, or at least a pretty tough abdomen. So the flies didn't seem to be able to actually oviposit in them. So we haven't ever been able to confirm that record from the literature. A thought, though, came to me, and I noticed that your last lecture was on the Asian giant hornet. It probably is a potential host for Apocephalus borealis because it's a close relative of yellowjackets. And you can imagine how many Apocephalus you could potentially get out of an Asian giant hornet if it indeed was a susceptible host. Hopefully we won't find out and the hornet won't become established in the Northwest. But if it does, there are plenty of Apocephalus borealis out there to perhaps help in controlling them. But of course, the big question is the zombie fly a bad actor in a bee movie, or is it an important player in honeybee hive losses? And that's something we've asked over the years. We initially put in a big grant to the USDA to carefully sample hives all over North America. Didn't quite get funded. Only about 8% of such proposals get funded these days, maybe less than that, actually, closer to 5%. And it had one reviewer who said, well, I don't know if there's enough evidence that this is an important phenomenon to fund it yet. And then another reviewer said, yeah, but if it turns out to be important, perhaps we should jump on it early. But it only takes one mediocre review not to get it. We were kind of despondent for a while, went and had a beer, talked it over, and decided that what we do is enlist citizen scientists and beekeepers to help us answer some of those questions. So what we did was start a citizen science project called Zombie Watch, playing on the media coverage that Apocephalus parasitizing honeybees had gotten throughout the world, literally, and using the common name that had been more commonly used in the media of ZomBees, spelling it in a slightly different way from zombie bees. And you can find us at zombiewatch.org. We're still there and still encouraging people to look for parasitized bees. We provide instructions on how to make some simple light traps to check for bees flying around at night. And you can see on a good night, you can get quite a few bees coming to a light trap. This was actually a light trap outside my house here in Glen Park in the city of San Francisco. Most of those bees were parasitized. And then instructions on how to transfer bees into containers, hold them for 10 days or so to see if maggots come out of them. And then we ask people to take pictures first of the honeybees, because we want to make sure that people actually know what a honeybee is. You're probably not surprised to hear that we get a variety of things turned into our site, including bee mimics, some of the bee mimicking flies, a number of yellowjackets, because people all saw cartoons when they were growing up on Saturdays that were black and yellow and looked more like yellowjackets than honeybees. So they think yellowjackets are honeybees, and that's fine with us because some of those yellowjackets are parasitized as well. And then we asked them also to upload pictures of the pupae. Normally we can tell if we get a good shot of a pupa, whether it's Apocephalus borealis or not, and pictures of flies if they emerge as well. So a number of questions we asked. How widespread is this parasitism of honeybees? We know that the fly itself is widespread in North America, from literally Alaska to Nova Scotia, with maybe an area in the middle of the country where there aren't any records. This shows what we knew in 2012 when we first started Zombie Watch. The blue indicators are museum records of the fly from throughout North America. The red ones are our records from the study we did at San Francisco State and published in 2012. Mostly in the San Francisco Bay Area, had one from Santa Barbara, one from Bakersfield. The Bakersfield one is a DNA sample that Joe DeRisi, a colleague at UCSF, did for us using his molecular techniques, and one from South Dakota, which also is from a commercial beekeeper sample that Joe had and that showed evidence of DNA from Apocephalus borealis in those hives. So this is sort of the base mark that we started back in 2012. We wondered whether this might be an emerging problem that we would see grow throughout the country, or whether it was something that had gone unnoticed for years. Or were there other reasons why honeybees might be out flying at night, and might we capture honeybees in other places that weren't parasitized? And finally, wondering if the zombie fly affected the behavior of its other hosts like Yellowjackets and bumblebees. I think this is about where we were the last time I talked to this group. This is from fall of 2013. At that point, we'd gotten a variety of submissions from citizen scientists across the United States, mostly at this point, and had gotten a cluster of new findings up in the Pacific Northwest, in Oregon and Washington, that increased that distribution of honeybee parasitism pretty much from Santa Barbara all the way up to the Canadian border. Today, we've now added the East Coast as well, with records from Nova Scotia, Newfoundland, Maine, into Ontario, Massachusetts, New York, and then down the Appalachians, essentially following the Appalachian Trail down into North Carolina, probably extending into to Georgia and into Tennessee as well, although we don't have records from there yet. We're hoping to get more submissions from those areas. There are a few areas where we know that the fly has been recorded before where we haven't found them in honeybees, although we've gotten a number of samples submitted. There are records from over here in Minnesota, nothing from there. There are records from around Salt Lake City. We've gotten samples from there, but none of them have turned out to be infected yet. And interesting, a lot of samples from LA County. We know that the fly occurs at least in coastal areas in LA County, but none of the samples from LA and honeybees have turned out to be infected. So that raises some more questions that need to be answered. Most of these new discoveries are made by beekeepers, often backyard beekeepers or small commercial beekeepers from around the US and Canada. A number of findings in California, Oregon, Washington, Vermont, Massachusetts, Pennsylvania, into Canada and British Columbia, Quebec, Ontario, and so on. And these are some of the samples that they take, some of the pupae that come out of those samples. And again, the pupae have a very characteristic look with these little nubs at the end and sort of concavity on the undersides that make them fairly easy to tell if you can get a good photo. Some of these citizen scientists became instant media stars, finding their portraits in local newspapers or on local websites, TV. Mark Hahn was the first beekeeper to find infected bees in the Seattle area. He was working for Microsoft at that time, and there were a group of Microsoft employees that kind of had a beekeeping club. They got involved in working with school kids and some of the local schools and bringing in samples and having them observe them. And indeed, those kids found infected honeybees in a number of those samples. Tony Cantrell from Burlington, Vermont is one of our better citizen scientists. He was the first person to find an infected honeybee on the East Coast in Burlington. And he also was willing to spend some time taking more samples. So he did a couple of years of sampling from his backyard bee hives of the intensity of parasitism of Apocephalus borealis on honeybees in Burlington, Vermont. This is not Tony. This is some other quasi-criminal in Burlington. So here's Tony's samples. This is from 2014 from his backyard. And he's looking at the number of pupae that he found in samples of his honeybees. So you can see it starts out about the first part of May, peaks in the middle of June, then goes through a kind of a low period and a larger peak then in late summer going into early fall. petering out at the end of October, which is, of course, when it starts getting really cold in Vermont. Not surprisingly, you don't find things happening at that point, that part of the world. And it looks like it's probably a two-brooded thing in that area. This is his do-it-yourself light trap that he designed and used for collecting these samples. Now we can compare that to what we found on campus at San Francisco State. You see kind of a similar pattern. Not much happening during the spring, building up in the early summer, peaking in the fall and early winter, and in the case of the Bay Area, extending on into January and sometimes even early February, and then going down again with very few infections throughout the springtime, coming up again and peaking again in the fall and early winter, crashing again and starting. So a similar pattern, but more of a unimodal, multi-generational pattern here in the Bay Area. Now, one of the things that we found through our sampling is that most of the hives that we've sampled in the San Francisco Bay Area are infected by the Phorids about this time of year, starting in September through October, sometimes earlier. Up to 80% of the samples that we get in those times show infection by the Phorid fly. So you probably have them in your hives, even if you haven't noticed them. And it's easy to overlook them because the infected bees mostly leave the hive and die someplace else. And you rarely find the pupae or the flies in the hive itself. We do have a few reports from citizen scientists, beekeepers, of pupae being found in their hives, but that's usually a hive that's not very healthy and on the way out, that probably doesn't have the ability to clean the pupae out and get rid of them. Sometimes we find Megacelia species in our Bay Area samples as well, so it's something you might be on the lookout for. This is the pupa of Megacelia gerardii, one of the numerous Megacelia species. This one found in the Bay Area along with Scaleris and others. This comes from a sample taken out of bumblebees that were being attracted to lights on the Google campus down in Mountain View. And you can see the Google beehives here nicely painted in Google colors. Since Google people are renowned for working late into the night, these lights stay on and the bees would fly out, bounce against the windows and the glass door there, and then die or wander around near those places. If you scoop up samples, get mainly Apocephalus borealis out of them, but you do get a few of these, we presume scavenger pupae, but it's also possible that that pupa came out of a living bee as well. And here's another one up here. You can see slightly more elongate, slightly larger pupa, the way that we can tell them. In May of this year, Frank Drummond from the University of Maine and co-workers published a paper on hives in blueberry areas of Maine, looking at varroa mite, tracheal mites. And I think just sort of as a lark, they decided to look for phorid flies as well. And the samples that have phorid flies in them are indicated by this yellow bar. And you'll notice that in 2015, all of the samples that they took, they had 9 hives or 9 areas of hives that they were sampling. Every one of them had phorids in those hives. And if you look kind of at the numbers, the density was comparable to tracheal mites and varroa mites in those hives. And they were surprised to find this, and it wasn't something they expected when they started out. And in fact, it's in some ways not too unexpected, because Apocephalus borealis was originally described from Maine. And the reason it's called borealis is because it's from the boreal zone, the boreal forest of Maine. That was back in the 1920s. It was only until a few years ago that we got a sample from Maine in honeybees. And now this study published in 2021, last May, that shows it abundant in hives, at least in blueberry areas of Maine. So another question that we've had is, since honeybees are moved around the world for commercial purposes and for backyard beekeeping, whether that movement might also result in Apocephalus borealis being moved from North America to other parts of the world. And if that happens, what kinds of places would be most likely to be supportive of them, to have the best environmental conditions for Apocephalus borealis to establish? So this is a collaboration with Eric Tihelka and colleagues at the University of Bristol and also some colleagues in China to try and assess that risk. It's going to come out in probably by the end of this month or so online. What we did was to take the known locations where Apocephalus is found. Here's the current knowledge of where Apocephalus is found around North America, and to generate a computer model of the environments that would be most conducive for Apocephalus borealis to survive. And you can see, as you'd expect, the places where we've gotten a lot of records turn out to be red. These red zones are the centers of best habitat for Apocephalus borealis. And then these areas are potentially suitable, maybe 50% possibility compared to the high possibility red zones, with kind of a break in the center of the country. Brian Brown thinks the break may be due to poor collecting across the expanses of Canada here, but it could be real because the environmental situation seems like it would suggest that there might be a break right along the 100th meridian that separates the eastern from the western US. But if you look into Europe, large expanses of Europe have habitats that would be conducive to Apocephalus borealis, as with the UK, large expanses of Asia, Japan, Australia, New Zealand, Southern Africa, parts of South America, parts of Mexico. So the potential habitats for Apocephalus borealis are many around the world. And we think that there should be some vigilance in checking hives that are moved into these areas from North America to make sure that they're not transporting Apocephalus borealis, not only because of what they might do to honeybees in these areas, but also what they might do to native bees, native bumblebees, and other bees that haven't ever experienced a fly like this before, where it could have a more major effect on their population biology. Okay, so citizen scientists have also documented other things coming to lights at night besides honeybees. Yellowjackets and other species of paper wasps have been turned in samples to us from both the east and west coast. Some of them have been infected by Apocephalus borealis, and that suggests that there may be something going on with the fly affecting behavior of these native hosts as well. We've got a few records of bumblebees, but not very many, given the frequency with which honeybees are infected by Apocephalus borealis coming to light, including one from Southern California and the Rancho Palos Verdes area, where we have records of Apocephalus borealis infecting honeybees, but we know that it's there because we found it in the bumblebee, or at least a citizen scientist did. We've also gotten lots of reports of honeybees coming to light from Florida, Texas, many from Southern California, many from Florida, a number from Minnesota, several from Connecticut, Las Vegas area, Phoenix, Tucson, even Malaysia, which are probably the Asian honeybee. These have turned out not to have produced larvae or pupae, so that we don't can't say that they were infected by Phorids. It makes us wonder if there's something else going on with these bees that gives them bee insomnia and has them flying out at nights and gathering around lights. Whether there's a more general response of honeybees to this disease that leads them to leave their hive and perhaps protect their hive mates from those diseases or stressors, and leads them then to be attracted to lights nearby if there happen to be lights and they happen to be flying around at night. Don't know, but it's an interesting phenomenon. Interesting that the literature on bees has very few reports of bees coming to light, honeybees coming to light at night. Some reports from Africa that suggest even foraging in some of the African honeybees, but in a report from Central America, a few others. Sometimes on bee discussion groups now, you'll see people talking about bee. So, as a beekeeper in the Bay Area, why bumblebees can exceed 60%, which is a pretty heavy hit. And even if those rates in honeybees are only half that, that's a rate that could contribute to hive losses. Now, they are hitting primarily foragers, we believe. So these are things, but as foragers are lost, they need to be replaced by other bees in the hive, and that could lead to a kind of cascade effect, especially if that's in combination with things like varroa mites and other bee diseases that we know are so common in the Bay Area and throughout North America. All the colonies that we worked with on the San Francisco State campus had varroa mites and a whole variety of bee diseases associated with them, deformed wing virus, et cetera, a trypanosomal disease, just a whole variety of things that were happening at the same time. And so this is pretty typical of bees in the Bay Area, which means that it's often hard to tell for sure what the absolute cause of death or cause of decline and failing of a bee colony is. And it's often probably a combination of effects, with varroa certainly being a top contender for why that happens, along with passing on pathogens. Interestingly, when we checked Apocephalus for pathogens, they turned out to test positive for a whole variety of bee pathogens. So potentially they could be vectors of pathogens as well as parasitoids of the bees. And then I wonder, the zombie bee is sort of the Rodney Dangerfield of flies in some ways. It doesn't get much respect. It hardly is noticed. Maybe we should call it the stealth fly rather than the zombie fly. Yet most hives in the Bay Area are infected, but most beekeepers aren't aware of it. And so I'd encourage you to be on the lookout for zombie flies in your hives. And it would be really great if you would participate in our sampling program and upload information to our website at zombiewatch.org. Best way is to use a light trap. Best time to sample is summer through early winter. Right now is not too late. Even last night would have probably been okay. We'd get bees flying on rainy nights and frosty nights in our sampling regime. So they can be out in times when bees should know better. Be great to have multiple samples over time rather than just a single sample. Even if you don't get positive results, we'd like to know that you got a negative result from a sample. And if you get a negative result, it would be nice to take some additional samples to see if that changes over time and if there's an increase or a decrease in the frequency of parasitism from year to year or throughout the year. So be on the lookout for adult phorids and pupae in your hives as well, especially in hives that are struggling a bit. And what can you do to keep your hives healthy and phorids away? I don't have a clear answer for how to keep phorids away, but probably the best way is to to follow the best practices that you've been doing to keep your hives healthy, controlling for varroa mites and other problems with the bees. A healthy hive is the one that's most likely to survive both the onset of phorids and other creatures as well. So I'd like to thank my Zombie Watch team. These are some of the members that have been part of the Zombie Watch group over the years. Brian Brown, who I mentioned is our fly taxonomist extraordinaire, who confirms all of our unusual findings. If we have a new record from an area we haven't seen them before, I'll send them to Brian to make sure that he agrees that they're Apocephalus borealis. Dragutin Perkovich, who is a person who was an instigator in getting this project going and a computer scientist that helped get his students involved in designing our website. Asim and Danius and others who were part of that. Chris Kwok, who remains to be an important person in the Zombie Watch team and continues studying bees and phorids in the Bay Area. Andy Zink, who's an insect behaviorist on the San Francisco State campus. Andy Core, who did some of the seminal early work, and Robert, who's been working with us for a number of years, who's on this, this Zoom get-together. And of course, special thanks to all the citizen scientists over the years who've shared their discoveries with us, including Sarah from BC, who is the first to discover Apocephalus-infected honeybees in Canada, and became a celebrity overnight on the Canadian news cycle. Okay, well, are there any questions, comments?
Remember, anyone needs to unmute to be able to speak. So if you have a question.
John, I might add one bullet point for beekeepers, and that's that in a heavy urban area of beekeeping, I've had to move at least 5 apiary locations because when these events peak, the neighbor, it becomes a major problem to close by neighbors. And so I've had to actually move 5 apiaries because of an overload of the phorids showing up. So that's one of the things that does affect beekeepers pretty substantially. And then the other thing is that it's one of those diseases that seems to just counting the numbers. So whenever you have good wing crawlers out in front of the hive, those tend to be the ones that are infested with Apocephalus borealis. And if they're bad wing crawlers, then those are the ones that are afflicted by mites. But by collecting the number of good wing crawlers, and you saw the number in that jar that I got, just counting how many I was losing per day, and the Yellowjackets would take them away. So you could never come home from work and count them at the end of the day. Yellowjackets took them for lunch and there was no evidence left. So most beekeepers just don't even get that there's a major problem, but I think it probably could knock out anywhere from 10, 15, 20% of the bees. And if you have other things that are knocking out 10% of the bees, that adds up to a substantial number that can really bring down, or bring up the impact to your bees. And so your hive would be the opposite of flourishing, and your honey harvest might be dropping because of lack of numbers of field bees. Anyway,
Yeah, good points.
I was just going to say that there are a lot of questions in the chat that you might check out and address some of them. Some of them you already talked about.
Okay, let's see. So I have a question about, are there single or several phorid larvae per parasitized bee? And the answer is it ranges from 1 to 25, I guess. So occasionally we'll get a single larva out of a bee, but usually there are multiple larvae. I suspect that there are bees that were attacked by more than one fly when we get things like 25 maggots out of a single bee, but we don't know for sure what's happening in nature. We've rarely observed the flies in nature, and as far as I know, no one's observed a bee actually getting parasitized, a honeybee actually getting parasitized in nature. Are bees not typically attracted to light? Well, bees are typically attracted to light. If if you were to bring a honeybee into your home, even during the day, its first response probably would be to fly towards a window where there was bright light coming in. So bees frequently are attracted to light. The question is, are free bees normally flying at night? And that's the difference here. The honeybees, one of the things I did when we were studying this and had a observation hive on campus is that I'd go in at night and I'd lift the frame so that you could see into the hive, and then I'd take a flashlight and move it around the hive. And mostly the bees ignored the light. These were the nurse bees and bees that were basically inactive at that time. But a few of the bees would follow it. You could follow them around. And so it's these bees that are active and alert, and for some reason flying away from the hive at night that then get attracted to the light. I think the light is just something that happens to be there and that attracts them. It's not that the bees are suddenly made more sensitive to light by the fly, but more that something is causing them to leave the hive at a time when they really shouldn't be. And let's see, I think Robert probably got some of this. I thought Walker bees had viruses. Often they do, but they can have a variety of things that are affecting them.
I saw from your map that Canada's climate is not very amenable to them. And I was surprised that Central America and South America is not very attractive of a climate for them either.
Well, the middle elevation, higher elevation parts of Central and South America would be appropriate ecologically. And the coastal parts of Canada seem to be pretty good as well. So Brian thinks that the Phorids don't like really hot, dry areas, this particular Phorid. But on the other hand, we have some samples, of course, from Walnut Creek and in Concord and places like that in the Bay Area where it gets pretty hot and dry during the summertime, certainly as hot and dry as it does in the LA basin. And there are examples of the fly being captured in places like New Mexico and the Great Basin Desert around Salt Lake City. So I'm not sure exactly what's going on. Also, the areas on the East Coast, are really typified by much more severe winters than the areas on the West Coast where we find the fly. And so that in some ways, I'm surprised that the computer program that generated those ecological niche maps, if you will, showed them to be as similar as they were. I thought it might show that there actually were two different populations that had different niches, one in the Western US and one in the Eastern US. But it turned out that that wasn't the case. It is found in Canada, though. We don't have very many records from Canada. Partly, of course, there are great expanses of Canada where there aren't very many people who live there, and perhaps not too many that have bees, although the bees are kept all the way up into Alberta and beyond, even up into Alaska. So there is the potential for people doing sampling in those areas. We've gotten some samples from Alberta, but none that have been infected with honeybees, even though there was a study done in Alberta back in the early 2000s that studied bumblebees and found a really high infection rate of bumblebees in Alberta by Apocephalus borealis.
I wonder, then if you're, so the bees dropping by the back door security light, if you swept them up, got them up and disposed of them, would you tap down the population in the area by removing the dead carcasses that had the larvae in it?
Sure. It's a good question. It's one that I've thought about. I think, it could help, but probably only a little bit. And part of the reason for that is that there are so many native hosts for the fly that, that's probably only the tip of the iceberg in your area. Even if you were able to, for a period of time, take all the honeybee hives out of an area so that there was no honeybee food source for the flies, they'd still probably do quite well on bumblebees and yellowjackets. So it's tough. It might reduce the numbers in the near vicinity to your hive, and that might be something that would cut down the numbers infecting your bees. But it's probably a relatively small effect. Some people have set out light traps just for that purpose, to trap the bees and then dispose of them to try and kill the parasites. There is something different though about this parasitism of honeybees and parasitism of bumblebees, if you think about it, because their hosts probably aren't going to respond in the same way in terms of population responses. So you typically think of a parasite host response as that a parasite builds up in number, its host goes down in number, the parasites go down in number, and then there's kind of a cycle through that process. But honeybees are a little different, because if a hive fails, it's usually replaced by another hive. And so, it's not as elastic a relationship as maybe it would be with bumblebees, where bumblebees numbers might decline over a year and then get hit by, then the fly goes down in numbers and bumblebees come back up, and flies hit them again and they go down, and thus they cycle through. In this case, beekeepers usually replace their hives, they split their hives, and pretty soon they're back to the same place they were, even if it's been a difficult time and a number of hives have been lost.
Well,
So, John, is it the case that if your hives are located in a place where there's dark sky and there's no artificial lights nearby that would attract bees, would those hives be less susceptible to these florid flies?
I think probably not. I had a colleague in Vermont who was going to try and do some sampling around hives in the hinterlands of Vermont where there were no lights to see if you would actually attract parasitized bees, but I don't think he was ever able to do it. He was setting out mosquito traps to look at mosquito populations in those areas. But it would be interesting to do some sampling of hives in more remote areas where they're not exposed to urban lights, and see what you would find. But you probably need to find the right area. I would guess if there aren't lights around, that the bees just fly until they run out of gas and die someplace off away from the hive, and that the coming to a light is kind of a coincidence of humans living nearby.
Well,
But-
some of those light traps have got other species other than honeybees, right?
Oh, sure. I mean, if you put it depends on where you are. If you put out a light trap in San Francisco at this time of year, you're not going to get much. You'll get a few small flies and some geometric moths coming in on some nights. But if you do that in southern Arizona in September, I mean, that light trap will be masked with all kinds of creatures, moths and beetles and flies and a whole variety of different creatures that are mostly nocturnal insects that then get attracted to lights at night. In fact, entomologists for years have used light traps as a way of studying insect diversity in various areas. But as a kid growing up, I always turned the lights on the driveway next to my house, and I'd collect insects at night there. I never remember seeing a honeybee coming to the lights in those cases. Did a lot of light trapping as a graduate student and as a professor with my classes. And, you know, I don't remember honeybees being a common part of those samples, but all kinds of other things for sure. Sometimes when we set up citizen science kind of projects with school kids, we'll encourage them to set out light traps and then encourage them to study not only bees if they happen to get them, but all the other things that come to lights. Because in some ways, this is a tough citizen science project because you're asking people to look for a relatively rare event. They need to be situated near probably a honeybee hive, either a managed hive or a feral hive, in order to have a good chance of getting bees. But they'll get a lot of other things coming in for sure.
So are you saying that it would not be normal behavior for the honeybee to go to a light unless they were infected or unless there's something else? Is that?
No, I'm saying it's normal for a honeybee to fly toward the light.
It is.
That's my,
Okay.
and I'm not a honeybee behaviorist, but most insects fly to light.
Okay. But
Yeah, most flying insects fly to light.
yeah, I thought you were saying that typically the honeybee is not nocturnal, so they wouldn't ordinarily leave the hive.
That's correct.
So if they left the hive, it would be because they're infected or something else is wrong?
Well, there's something going on with them.
Okay.
Yeah, it could be everything from a raccoon or a skunk or a bear to Phorids, I suppose. But in most cases, there's no obvious reason that they'd be leaving the hive.
Right. Right.
Other in this case, other than having something going on inside of them.
Yeah.
I think there could be certainly given the number of honeybee records now that we have from people finding them at lights that are not infected with Phorids, that there must be a number of reasons why honeybees leave their hives at night. But you know, it's not a normal behavior, and certainly not a normal behavior for them to be flying out on rainy nights or frosty nights or things like that.
Right. Okay. Yeah. A few years back, I had, you know, a series of weeks where the bees were attracted to a side light.
Mhm.
And in the morning, most of them were dead on the ground there.
Yeah,
So,
that's pretty typical.
okay.
There is a paper from a number of years ago. I think it was done in Illinois during the summertime, that where screens were put on the hive in the morning, and then some bees were trying to come back in. So they'd obviously probably been stranded someplace and didn't get back to the hive at night, and then were trying to come back into the hive during the day. But you know, I really don't know of any careful studies that have looked at bees being active at night or being attracted to lights at night, other than the stuff that we've done.
Okay. Thank you.
And I wouldn't characterize ours as a careful study of exactly what's going on in the bee itself, but it's sort of a initial documentation of a phenomenon that needs explanation.
Right. All right. Thank you.
So it's puzzling to me that the phorid fly infestation should peak in the late summer and fall, especially when honeybee is a host that's available 12 months of the year.
Yeah.
Why should they shut down at the end of winter and stay down until, you know, a rather bad time of year?
Yeah, great question. I don't have an answer for it. If I were if I were a fly, I'd want to be active in the spring when, you know, bees are increasing in numbers. There's a lot of nectar flowing. That's true both for honeybees and for bumblebees.
Is it perhaps that they have a preferred host, and only when that preferred host dies out, that they'll go after the honeybees?
Well, it's certainly true that bumblebees go down in the fall. So their numbers diminish in the fall and they overwinter as queens for the most part. So that would fit. We haven't consistently found high infestations of Apocephalus borealis in bumblebees during the spring, though, to fit that pattern.
Like they're hitting them at a stressful point in their cycle.
Well, it could well be that stress is part of it. That's certainly the most stressful time for both of them, for honeybees and for bumblebees. I mean, many times it's the end of the life for worker bumblebees, and it's the most stressful time for hives as well going into the winter. So it could be that it's that being able to overwhelm bees that might be better able to protect themselves, fly faster, scrape off a fly more easily, whatever it might be. Could also be that are closely associated with, more closely associated than we know with Yellowjackets, which tend to, as you've reported, peak during the fall. And that's part of the cycle as well. But I don't have an answer for it. It doesn't make a whole lot of sense to me just looking at resource availability for the flies.
I wonder if Randy Oliver could develop a grooming behavior for the bees.
Maybe so. I mean, you have to be able to kind of reach your hind leg back over your abdomen.
No, each other. They could do each other.
They'd have to do each other. Maybe have to do group foraging or something. We assume that this is happening on flowers primarily, but again, we don't know for sure. We have captured Phorids, adult Phorids around flowers, but the number of observations are small. So there's still so much to learn about what's going on with this system.
John, do you have any idea what the incubation time is between when the egg is laid and when the larva hatches out and probably begins to, actually feed on the bee?
Chris Kwok has done most of the rearing work on these things, and I think he would estimate it between a week to 10 days to complete that cycle, probably closer to a week. From egg laying to coming out. So what that would mean is that when the flies are, when the honeybees are actually leaving their hive, they have very small maggots in them. And obviously they couldn't fly if the maggot had eaten out their wing muscles. So they leave their hive at a relatively early stage of parasitism. And then it's something like 5 to 7 days after that, typically the maggots emerge from them. So it's probably about a week cycle from egg to maggot to pupa, and then another month or so before the flies emerge.
Wow.
Which is a strange thing too. I don't understand why does it take a month for this little fly to metamorphose in its pupa? It seems like it could turn around a lot faster than that. And in fact, there's an earlier paper on Apocephalus and Yellowjackets from the East Bay there that reported the flies coming out much faster than that. But for us, typically it's more like a month.
So they're probably
I think
not,
I was
sorry.
Oh, it's Robert. I was whether I was putting them in individual glassines or just in a jar, it tended to be by what I was getting, about 5 days for the pupae packets. I mean, for the larval packets, and then another 5 or so days before the flies actually hatched out.
Yeah, it's probably temperature related.
Yeah, that's probably
where
true.
they are
Absolutely.
in terms of especially the pupal development. And one thing we don't really know also is where these things are pupating in nature. I mean, we assume that they slither away from a bee and go into a crack in the ground or something like that, which is sort of the typical behavior for things like that, but haven't actually observed them. I did find some pupae outside my house under some plants growing through cracks in the concrete. So that's my one observation of pupae in urban nature of San Francisco. But what would happen in an open field or some place like that is unclear, and how variability in those habitats would affect survival and generation time.
So in a cold climate, things freeze, and apparently freezing doesn't kill them off. Do they pupate to get through winter? Do adults, are they survivors through winter?
It's probably the pupae. The adults don't live very long, so it's not likely that they're the survivors. And there was a study that put pupae into refrigerators and basically held them for periods of time and then brought them out and they emerged. But exactly where they are in nature, in places like Maine or Alaska or Alberta or places like that where it gets really frigid, it's hard to know. I suspect that they pupate down in the ground, and snow cover and things like that keeps the temperature that they're in to around 32 degrees or so, rather than being super cold. But again, nobody's actually observed that or seen it.
And John, there's no evidence of infestations causing bees or hives to become more aggressive, is there?
Not that I'm aware of.
Okay.
That's a question I get sometimes, whether these bees are more aggressive, whether someone needs to worry about being stung by them, and whether if they get stung, whether you will become a zombie. That's happens maybe in young adult literature or something, but not in the real world here. But it's a good question, because potentially it could affect their behavior, but I don't. For the most part, the bees are pretty much whacked out. They don't seem to have the energy to be too aggressive at that stage.
So you haven't described this experience that I've had with bees coming to light at the back of my house. But I, for a few years ago, would have bees come to the back light. And then if I opened the door, they would come inside. And then they would exhibit the most outrageous behavior, where they would skitter across the kitchen floor and back and forth really fast, and maybe flying a little bit, but very odd behavior. And then then they would die very quickly.
Yeah. Well, I think maybe the behavior you're describing is something a little earlier than the behavior of the picture I showed of the bee kind of wandering around in circles. That bee was near the end of its frantic time, and maybe you were seeing them from in the early part of frenzy to the end.
Yeah, so I'm not sure when in the infection period do they act like that. So, when I did capture a few and put them in a jar, I didn't count the number of days until they hatched out, but they did have phorid flies.
Yeah, yeah.
So they're actually impacted earlier, soon after the flies lay their eggs.
That's right. And I spent a fair number of hours dissecting bees that were under light to see if I could find the larvae or eggs or whatever would be in them at that time. And what I'd find were really tiny larvae, usually sort of up in the dorsal portion of the abdomen, sort of just before they're going to have to push through and get into the thorax. So I think typically the bees that are flying out have newly emerged larvae in them that are probably only a day or so old, maybe a little older. The study in Maine that I mentioned, they actually counted phorids by dissecting the bees rather than rearing them out. So they counted eggs and larvae that they found inside the bees, which is really quite a task to make sure you find all of them, because they're quite tiny. Once they get large and are feeding up in the thorax, it's real easy. You just break open the thorax, and they're easy to see and easy to count. But when this whole thing starts and they leave the hive, they're quite small.
Well, like Robert, I was, very concerned about this because I've been working on, responding to neighbor complaints. Luckily, I had a good relationship with my neighbor, and the same thing was happening to them. The bees were going to their lights and going in and acting really crazy. But your light trap, information was extremely helpful at that point. And once I put the light traps out closer to the hives, really close to the hives, I never had any problems with them coming to the back door lights anymore.
Yeah, you got them early.
I guess so.
Yeah, maybe so.
But that
So
suggests
that's
that
a
they're-
good observation, because one of the questions I have on the chat is, how close should the light traps be? And so I think you want to sort of have a balance. You don't want to have them so close that if it's a bright light trap, that it's interfering with the behavior of the bees. But within a couple of meters or so is probably not bad.
Yeah, I had it in the apiary, not right next to the hives, but I wanted them to go to that light before they went to a house light.
Yeah.
And I didn't use a very bright light. I used a smaller light. They did come to it.
My observation is that they seem to be attracted to a variety of different lights. We tend to use UV bulbs that or fluorescent bulbs that have a fair amount of UV associated with them, black light bulbs. But I've seen them come to sort of LED flashlights, and white light flashlights have virtually no UV coming out of it. The LEDs there, or LED flashlights that are UV, they'll come to those. It doesn't seem to require a lot of bright light necessarily to attract them.
So that suggests also that because trapping them was efficient, that there weren't very many native hosts in the area to source the thousands.
Well, it could be. It's interesting with Yellowjackets, or the native hosts that have most commonly been reported at lights. And, they also are the species that often nest near human dwellings, especially the ones that nest up in the trees or in eaves of houses. So they may start off being closer.
And John, you said that they normally won't, the flies actually don't go into the hives directly, that they're actually infesting the bees outside of the hive?
That's what we believe. We spend a lot of time staring at hives looking for small flies. I used to go out and collect small flies off the side of the hive that Gretchen had on campus. And I found a variety of things on the hive. It was actually quite fascinating. But I never saw Apocephalus on the hive. Now, Chris Kwok has told me recently that he's seen Apocephalus near some of the hives he's been keeping. So it may be that it happens sometime, but it doesn't seem to be the most common way in which they infect bees anyway. Again, if I was a fly, I would come out in the spring and I would hang out around honeybee hives. But that doesn't seem to be what they do.
So, you know, the way it would weaken a hive would be to retard the resources that the hive can bring in, basically because it's foragers that it's affecting.
Yeah, I mean, the effect on the hive is loss of foragers primarily. And the effect that that has on the dynamic of the hive itself is, there's the sequence of responsibilities that worker honeybees go through. And if you remove foragers, some of the nurse bees are going to have to become foragers before their time, if you will, in order to fill in.
It definitely picks it up. Yeah, that's interesting.
And that can lead to some problems internally in the hive with feeding brood and cleaning things out, and etc. At least that's our supposition. That
So, John, are you actively still? Is the university still actively researching this, or is it not?
well, we're still running the citizen science project.
Okay.
And so that's still active and going. I must say I haven't been on campus since February 2020, and you can imagine why. The campus opened up again this fall, but I haven't sought permission to go back on campus yet, and a lot of the areas are still controlled because of COVID concerns. Chris had some hives on campus. He's now moved them off campus. Again, because of issues of being able to go on and take care of them.
Yeah, I know you've been working with Robert as well, but
yes,
I'm also
Robert's been great.
I'm also a commercial beekeeper, so if there's something that we could do to help you guys, just reach out.
Sounds good. Well, we're still interested, and there's some of the sort of difficult questions of what percentage of foragers are infected in a particular hive, and that requires sampling a certain number of bees from the hive in a standardized way and so on, and it becomes difficult because of the timing of all this. So the big time for the phorid is fall and winter. And the most stressful time for the honeybees is fall and winter. So it's not a time when beekeepers normally would like us to open up a hive or to remove foragers, etc., from a hive. And it makes it difficult to get large samples and figure out what's going on. But certainly some of the samples we have taken on campus get up in the 20% plus range. And the trying to remember off the top of my head, the main samples I think got up between 10 and 15% of foragers infected in June, which probably wasn't the prime time. If it's similar to Vermont, prime time would have been September. And I would predict the infection rate would have been higher.
And do you guys have a protocol for taking samples? If you're looking to take samples from a production hive?
What we did on campus is that we would sample bees as they came back to the hive and sample bees as they left the hive, and take a sample of 50 and literally collect them as they came in and out of the hive. Not exactly randomized, but not targeted to any particular bee. And then we could compare the parasitism rates of those that were leaving versus those that were coming back. And generally the rates were higher in those coming back.
did you just wait for a pupa to, or a larva to hatch out, or did you dissect them? The 50?
We isolated them in individual glassine envelopes or vials,
Okay.
and then waited for the pupa to come out. So it's fairly simple bioassay. Now, that also means we may have missed things where the larvae didn't complete for various reasons. And we know that a lot of samples that are collected, the pupae don't actually hatch. And that's probably due to the conditions that they're kept in. And it may well be that, I mean, in some cases, the larvae, for whatever reason, don't hatch out of the honeybee. I mean, they may get caught with diseases or some other thing on the inside of it as well. So what I'm saying is that we probably have a minimum estimate rather than a maximum estimate using that. The other way to do it is now to use molecular probes and to test for DNA of phorids in individual bees. I don't have the resources at the moment to do it. Joe DeRisi at UCSF was involved in that kind of sampling with bees from commercial beekeepers back several years ago. I think mostly he's involved in human virology right now, though, rather than honeybee virology.
So I have a question on the, I don't know how I can elaborate on, you just mentioned that you're getting samples from the forages coming in and out. But first question is, all my questions related to the treatment of the hive and all different treatments that we have. Would they affect these parasites at all? So first question is, when you're collecting samples, was there any were there any treatment in place in the hive? And if you do, what do you have? And the next question is then, there are some of a lot of it's like I was going to ask Robert specifically. He's in a diehard oxalic acid treatment, and do they affect and how they affect. And then furthermore, there are a lot of different treatment, thymals to farmazan and to Emeritus. And then Emeritus is like a small amount of doses on it. Not killing and disabling the mites, then would they affect these parasites at all?
Great questions. The answer is I don't know. That's not something we looked into. The hives that we had on campus basically were untreated. I think maybe Gretchen put some powdered sugar on in there occasionally or something, but that was about it. There was no oxalic acid or miticides of any sort that were used. And, you know, those hives, as I mentioned earlier, were positive for all kinds of bee viruses and other bee diseases, as well as for phorid flies and varroa mites. And in fact, the hive seemed to survive and prosper for a number of years with relatively little management. On the other hand, it wasn't robbed for honey either. So the honey stores were probably pretty stable for the bees, but there wasn't any treatment in place. And we haven't looked at the effect of various treatments for mites and so on the phorids. It would be interesting to look at, and it's certainly possible that there might be something going on there.
Okay. Well, this is really eye-opening. This is the first time I've encountered the questions and these parasites at all. So I have about 7 different sites. I might start putting on some different lights and so forth, and by the different treatment and the results of differences between the bees are collecting and whether they have that and how much and what does. And probably I'm like to concentrate next year, 2022, and part of the project, see what's coming out of it, and it would be really interesting to see. Thank you for
that would be really interesting to do. So
Yeah.
I would really be interested in what you might find out.
Okay, sure.
So one partial answer for you was that one site that I get feedback from people who live there, and there are also some skylights fairly close to the hives. So that's one of the attractants. And then they also have a porch light. So if there's anything unusual going on in terms of numbers, they tend to let me know. About a month ago, I ended up putting refreshed oxalic sponges in.
Mhm.
And there was a pretty huge wave of dead bees that were attracted to the lights after I added the oxalic. So to me, it's kind of it resembled when I would lose queens to formic, and they would be the older queens that would be having issues. So maybe they were just weakened, and then the oxalic really, kind of dropped the hammer on them in terms of accelerating their end of life. I don't know.
Mm.
But I did get a call from the landowner kind of expressing major population around lights. So oxalics seem to have an effect.
I mean, I think I should, because they're small. I mean, the creatures are so small, and it wouldn't affect. My whole point is that all the treatment doesn't affect the bees, but it affects the mites, and then the parasites are small enough to it should affect them. So it'll be really interesting to see it. I'm gonna keep my eyes open on that.
I have one more question here. It's a question about having recycling bins and things like that around, and whether that would be a place that would propagate the flies. And the answer to that is not Apocephalus borealis, but it might well be a place that propagated other kinds of scavenger phorids. Megacelia and other scavengers certainly would love any kind of decaying substance, be it leaves or animal decay or excrement, as a place to develop. But it would not be a place for these particular flies. They're specialized parasitoids that develop inside of honeybees and bumblebees and things like that.
Well, if that was really the last question, I'd kind of like to take the opportunity to say thank you, John, for the work you do. And for you, it's 14 years later, and you're very persistent and continue to push on this as an issue. And it's just, there's very little reward because nobody will take this seriously because it doesn't kill off the hives. And so to have you continuing to work on this and get the word out is so appreciated by me as a beekeeper and citizen scientist. So thank you for all you do.
Thank you, Nikolai. Appreciate it.
That's Robert in his disguise, by the way.
I came in on the
also known as.
I came in on the laptop also, so.
Okay.
And he's tricked us a few times that way, so.
Yeah, he's a trickster.
All right, is there any other questions for John?
Hearing none, I would like to thank you again for coming to give us your presentation, and I wish you all the success in your retirement and in your continued pursuit of the Phorid fly.
Thank you.
Yeah, thank you. Thanks for the invitation. And maybe in a few years, we'll have some additional information, especially if some of you get involved with some of these interesting projects of looking at effects of miticides and other things. It'd be really neat.
All right, well,
Thanks again.
That was a great presentation. Appreciate it.
Thank you, John.
You're welcome. Bye.